And we'll start with Dr. J.P. Clancy, Vice President of the Clinical Research CF Foundation.
Thank you very much. Can everybody hear me okay? Just make sure that's working correctly. Sorry, just in terms of conflict of interest, exactly the same as those highlighted by Mary Dwight as a employee of the CF Foundation. So first of all, thank you everyone for this opportunity. My name again is J.P. Clancy. I am a physician. I'm a Vice President of Clinical Research at the CF Foundation, and I've been a professor of pediatric pulmonary medicine at Cincinnati Children's Hospital Medical Center and also at the University of Alabama at Birmingham. I've been involved in the care of CF patients and also involved in CF research for more than 25 years. And that includes studying CF in the laboratory and also being part of clinical trials of new therapies. In short, oops, I lost my my notes. 1 second here. There we go. There we go. Sorry about that. In short, though, I think I can say that I've spent my career really focused on CF care and research. For decades, we've been only able to offer CF patients therapies that treat their symptoms. That changed dramatically in 2012 with Kalydeco. This was the first therapy, CFTR modulator, that targets the underlying cause of CF. As you probably are aware, modulators act at the cellular level to substantially delay progressive and irreversible organ damage caused by CF. And clinical data indicate that modulators can truly be transformative, improving and extending the lives of CF patients. Highly effective modulator therapies, particularly Trikafta, demonstrate dramatic benefits compared to existing therapies across key clinical outcome measures. Although each modulator provides significant benefits to eligible patients, Kalydeco and Trikafta demonstrate such a high magnitude of benefit across numerous CF manifestations that we consider them highly effective modulator therapies, or HEMTs. These produce dramatic benefits compared to existing therapies across key clinical outcome measures like lung function, growth, risk of pulmonary exacerbations, sweat chloride concentrations, and quality of life. Clinical trial results show that patients on Trikafta with only one of the most common CF-causing mutations, F508del, demonstrated a 14% average improvement in lung function compared to those not on a modulator. A clinically significant improvement is felt to be around 4 points. As also noted, it also reduced pulmonary exacerbations by 63%, improved weight, and dramatically improved respiratory symptoms. Similar benefits were seen in those with 2 copies of F508, but the study design was different. These improvements translate into real-life benefits described by patients, benefits that are really difficult to quantify with the data set. I think it's also important to note that access to Orkambi and Symdeko is still critically important to treat the underlying cause of CF. Modulator therapy responses vary from person to person. The availability of multiple therapeutic options ensures that patients who do not have a strong response to or don't tolerate a highly effective modulator have access to other modulators. They are also essential for preserving lung function and other organ function in children who are not yet candidates for Trikafta. Modulators have the potential to dramatically alter the course of this chronic life-shortening disease, particularly for those who start treatment at a young age. Patients starting modulator treatment early have the greatest potential to benefit long-term. We believe early restoration of CFTR protein function will have numerous beneficial downstream consequences, such as preventing organ damage and halting disease progression. This preventive use could lead to a fundamental change in how we care for patients, like using symptomatic therapies less frequency and reducing treatment burden dramatically. I believe that patients treated with modulators from early in life would have lifespans very similar to the general population. Just a decade ago, this was a dream. Data also suggests that long-term modulator use will fundamentally alter the course of CF. Because CFTR modulators restore mutant CFTR activity, all of the downstream consequences of lost CFTR activity can be prevented, reducing the need for future chronic therapies. The CF Foundation is leading the PROMISE study, which will include patients 12 and older on Trikafta to understand its impact on all facets of CF, including patient-reported outcomes, quality of life measures, and other organ impacts. This study will ultimately extend to younger patients pending FDA approval. ICER's review of CFTR modulators so close to the approval date does not really allow enough time to collect long-term data. And without these data, these therapies might be significantly undervalued. Long-term data are essential elements of any model used to assess cost effectiveness. Trikafta received FDA approval about 10 months ago. There are concerted efforts underway in the CF research community to understand the long-term and real-world impacts of modulators on health status, quality of life, healthcare resource utilization, and other factors. By not including these results, this analysis significantly undervalues Trikafta. In closing, I wanted to note that CFTR modulators have great potential to dramatically change the trajectory of an individual's life. I recently heard from a young adult patient that I cared for 10 years ago. She's been on Kalydeco since its phase 3 trial in 2011. She's now in graduate school. She maintains lung function in the 90s. She needs IV antibiotics about once every 2 years. Generally takes only Kalydeco and enzymes to remain well. While this is an individual instance, her experience is a glimpse into the near-term future for many with CF treated with highly effective modulators. Thank you so much, and I'd be happy to answer any questions if time allows.
Thank you for your perspective. I'll first ask if any of the CTAF or ICER presenters or staff would like to respond to anything they've heard. And of course, others on the panel, you're welcome to raise your hand. Okay. Well, again, thank you for that perspective on the existing and potential benefits of the modulators. We'll now move on to our next speaker. This will be Siri Vaeth, Executive Director from the Cystic Fibrosis Research Incorporated.
Good morning. Can people, I don't know if my camera is on. Is it working? Can people hear me? Yes? Okay, great.
Yeah.
Good morning, esteemed forum members. I have no other changes aside the disclosures on that slide. I address you today on behalf of my daughter, who lives with cystic fibrosis, and on behalf of Cystic Fibrosis Research Incorporated, CFRI, also my organizational peers in the CF Engagement Network, and the thousands of members of the national CF community that we serve. We are deeply concerned by the conclusions reached in ICER's evidence report regarding Trikafta as a CF therapy. While our organizations are listed as stakeholders whose input impacted the report, we reject its conclusions. The devastating impacts of cystic fibrosis cannot be fully conveyed until you experience them or you watch your loved ones suffer them. Last year, half the individuals with CF who died were under 31 years old, largely from respiratory failure. Diabetes, sinus disease, pancreatitis, osteoporosis, liver disease, diabetes, male infertility, chronic fatigue, chronic pain are additional complications. Advanced lung disease may cause a chronic sense of being smothered, and the fear of a catastrophic hemoptysis or pneumothorax looms large. Life-extending transplants are fraught with their own tremendous risks. The report minimizes the pain and suffering by those experienced with the disease. We are opposed to ICER's use of quality-adjusted life year or QALY measures. This discriminatory methodology will always penalize individuals with disabilities, and specifically those with incurable chronic diseases. The concept of the QALY was developed specifically to ration medical care and has nothing to do with patients having access to the most appropriate drug for their condition. The use of QALY-based thresholds to determine benefits and coverage is banned by the Affordable Care Act and Medicare, and likely violates the Americans with Disabilities Act. The use of QALY is especially problematic with CF due to the many CFTR mutations and varied disease expression. The utility weights used to determine a person with CF's QALY based on FEV1 are simplistic. To have a more accurate measure would require a patient-centered analysis that includes a diverse list of disease symptoms. By placing a numerical value on one's quality of life and health utility, you've taken a disturbing step towards placing greater value on the healthy and lower value on the disabled. And we don't agree with the claim that ICER's use of quality to determine drug price value is not inextricably related to placing a value on the lives of those needing the drugs. We believe the report's economic analysis leads to an overprediction of medication cost over time. And yes, the quote that people keep coming back to, when it's evaluated as a curative therapy, and was found to exceed cost-effectiveness thresholds, even under the assumption that it maintained individuals in normal health. In light of the robust CF therapeutic pipeline, we question the use of static pricing with projected lifetime use of Trikafta. Other companies, we are watching closely. They're developing CFTR modulators, mRNA therapies, genomic editing techniques. It is unlikely Trikafta will be the only option in the not too distant future. We remain concerned that ICER did not assess the full array of medical costs associated with CF and its complications. The analysis also fails to include projected loss of income caused by the disease for adults with CF and CF caregivers. A therapy that reduces medical costs while enabling people to work has far broader financial implications. And of course, we cannot put a price on the savings in physical and emotional suffering. Who knew that shortly after Trikafta's rapid approval and availability, we would be facing a pandemic? I've spoken to individuals with CF who had COVID who strongly believe they survived thanks to Trikafta. A woman with end-stage CF was literally prepped for transplant surgery in the hospital when it was determined that the donor lungs were not viable. She was put on Trikafta prior to its approval to buy time until new lungs arrived, and soon after was well enough to be removed from the transplant list. We can analyze the economics of that and put a number on it, but why? There's no price or number that could sum up the impact of Trikafta on her life and those who love her. We believe that despite input from patients and advocacy groups, the value of Trikafta is not accurately conveyed, potentially giving credence to payers who seek to reduce costs by denying coverage for this drug and other new therapies. We have sincere concern that should this flawed analysis be embraced by payers to deny access to these therapies, there'll be a chilling effect on innovation. It is a unique challenge for CF and other rare disease groups to entice biotech companies to enter their realm. Approximately 10% of those with CF are unable to benefit from these therapies due to their mutations, and only 7% of the 7,000 rare diseases have approved therapies. It is difficult to incentivize research and drug development when the numbers who will benefit from the drugs are so low. And for those without new therapies, facing an early death remains a tragic reality. ICER very, very rarely finds rare disease drugs to have adequate cost-effectiveness ratios. We fear that this consistent non-patient-focused assessment will serve to suppress research and discourage investment in drug development. This would be catastrophic for the CF community and other rare disease groups. We strongly object to the methodology used to evaluate this therapy and as such reject the report's conclusions. Thank you.
Yes, thank you for providing your thoughts on both the qualities and the use of cost in general. I'll go again to our CTAF and other presenters if they would like to respond to any questions. And then if not, I think we can start maybe with David Ryan. David, would you like to respond?
Sure. Just looking to see whether I show up on the screen here, but I was going to comment that both the QALY and the EVLYG are ways of looking at benefits, and the QALY really does show the dramatic benefits in quality of life that were found in the trials of Trikafta. And those benefits, as Dr. Tice was pointing out, are dramatic. This is a great drug. It has a huge benefit on quality of life, and our modeling suggests decreases dramatically in lung transplant. It suggests extension in life. But even when we assume that Trikafta does something it's never been shown to do, which is give somebody with CF a normal life if started at birth, we still end up with these extremely high cost-effectiveness ratios. And that has nothing to do with the value of the life of somebody with cystic fibrosis, that has to do with the price of the drug. And that price wasn't handed down as if from on stone tablets that it was unchangeable. That was a price set by the manufacturer. And it's that price that the QALY and EVLYG are raising concerns about, not the ability of the drug to be helpful and not the value of the lives of people with cystic fibrosis.
Thank you. I think at this point, just in the interest of time, we're going to move on. Again, thank you, Siri, Vaeth, for your presentation. Our next speaker will be Juliana Keeping, who is a parent of an individual with cystic fibrosis.
Yes, thank you so much. To take myself off mute and on video. My name is Juliana Keeping, and my son Eli is 7 years old, diagnosed with cystic fibrosis at 2 weeks old. I have no conflicts of interest to report. Thank you so much for having me today to speak on behalf of myself and my son. Eli has the most common version of CF. He takes Symdeko to treat his illness, along with other drugs, inhaled treatments, and physical therapies. Eli's among the 90% of people living with CF for whom our latest modulator, Trikafta, can make a difference, potentially a big one. Yet, 10% of the estimated 100,000 people in the world with cystic fibrosis living with even rarer versions of my son's genetic life-threatening illness have no modulator. That's why my family remains dedicated to finding treatments and cures for every person with CF and continues to fundraise for research as our community has for decades. Trikafta gives me hope that one day my son will not only be able to breathe like me, but to outlive me. But consider that Trikafta costs nearly $312,000 per year. This is a drug that treats the vast majority of individuals with a rare disease. What then would an acceptable price be for a drug that treats the remaining 10%? It's a question that should give us pause. Drug makers like Vertex take advantage of patients like my son by pricing drugs not on what makes a fair and reasonable return for their investment, but what they can get away with. Today, Americans with cystic fibrosis are being forced off of the very modulators that they fundraise to create. Drugs don't work if people can't afford them. My son could just as easily become a victim of unrestrained drug pricing as he could his disease. The bottom line is that our modulators cost too much. ICER's 2018 report concluded the prices of Orkambi, Symdeko, and Kalydeco, which range between $272,623 and $311,701 per year, would need to be slashed by about half to be considered cost effective. I agree. So does the latest draft report, which also states, and I do not quarrel with this conclusion, that a discount of up to 66% would be necessary for Trikafta, which again lists at nearly $312,000 per year to be considered cost effective. My reasons for concluding the prices are too high are different from, but they complement ICER's analysis. As patients and loving caregivers, we must stand guard against unjustifiable price gouging whenever it arises. The CF community's charity dollars and taxpayers played an outsized role in the creation of our modulators. The pharmaceutical industry wants to compensate investors for the high risk involved in bringing the drug to market. Without massive price tags, new life-saving treatments will not be invented and made available to people who need them, or so they say. But in the case of all CFT modulator drugs, the CF community and taxpayers took the early critical risk by investing money into the research and clinical trials. Vertex moved to acquire the IP only after the treatments were shown to be viable. And today, Vertex continues to exploit its monopoly to gouge patients and payers. It says it needs to charge these prices to continue to invest in new drugs. Yet Vertex has, since 2018, relied on a tax bonanza to repurchase hundreds of millions of dollars of its own shares, money that could have been used to lower drug prices. So far, its stock buybacks total more than $500 million, with another $464 million in stock repurchases authorized. Leaders have bragged about having billions of dollars of cash on hand. The Journal of the American Medical Association found there is no evidence of an association between research and the development costs and prices. Rather, prescription drugs are priced in the United States primarily on the basis of what the market will bear. Truer words could not be spoken about Vertex pricing for these drugs. Patients cannot afford these drugs. Robert Davis, he's a 50-year-old Texan living with cystic fibrosis. He was forced off his Vertex modulator because he could no longer afford it. Faced with $17,500 in drug costs, Robert took 1 modulator per day instead of 2. Within weeks, he began coughing up palmfuls of blood and was rushed to the hospital, where he stayed for a week, followed by another week of IV antibiotics at home. Patient assistance programs never cover all his costs because the unrestrained list prices of his drugs are too high. High drug prices are dangerous for people with CF like Robert and my son Eli. We are in the midst of a global pandemic in which millions of Americans are losing their jobs and employer-based insurance. People who lose private insurance will increasingly face the full brunt of their drug costs. They will
Sorry, I'm going to have to ask you to please summarize since we're over time.
I will very quickly say they will discover copay programs run by the drug companies only work if those companies can charge extreme prices to their insurers. In conclusion, Trikafta's price should be lowered. Orkambi, Kalydeco, and Symdeko are priced too high according to ICER's analysis. Vertex does not deserve a high-risk adjusted price as a philanthropy and taxpayer-funded research lowered the risk for the company dramatically. Vertex could use its tax windfall to lower drug prices, but it is instead paying executives and buying back stock. It can easily lower the price to come in line with ICER's findings. Thank you.
Yeah, thank you for providing your direct and personal perspective on this. I don't see any questions, and given that we are a little bit short of time, I think we'll move on to our next commentator, which will be Laura Rogers, who is also a parent of an individual with cystic fibrosis.
Laura's having a little trouble dialing in, and so while we're waiting for her to dial in, why don't we move to Riedy, and then we'll turn to Lori after he's finished his remarks.
Okay. Do we have Chad Riedy, National Advocacy Chair?
Yes, I'm here. Good afternoon, and thank you for the opportunity to share the impact that Trikafta and other CFTR modulators have had on my and my family's life. My name is Chad Riedy, and in 1984, at the age of 3, I was diagnosed with cystic fibrosis. Upon my diagnosis, my parents were given the news that they shouldn't expect me to live to see my 12th birthday. I am now 39 years old, living in Alexandria with my wife, Julie, and our 2 boys who are 10 and 6. It is hard to imagine that at the time of my diagnosis, the CF gene would not be discovered for another 5 years, and that it would be 10 more years until the first targeted therapy would be approved. Now, 36 years later, we have moved beyond simply managing the symptoms and damage caused by infections to actually being able to treat the underlying defect in the gene. You all have seen the data that demonstrates the impact these modulators, specifically Trikafta, are having on lung function, hospitalizations, and exacerbations. But this is not the whole story. Just by looking at its impact through data and quality adjusted life years, you cannot hear the laughter in the faces of my boys as we run around together. You cannot hear the joy in my wife's voice as we take a walk and actually have a conversation while doing it. You cannot feel the elation in my heart as I carry full laundry hampers up 2 flights of stairs without stopping. You cannot see the sweat that drips from my forehead as I carry and pour 50-pound bags of sand for our new walkway and patio. You cannot see the tears in my family's eyes as our world opens up with possibilities. While these might not be a big deal to many, they are to our family. It was just 5 short years ago when my lung function dropped to 24% because of an infection in my lungs. I could not pick up my boys and hold them when they got hurt or carry them upstairs. I could not walk 10 feet across my living room floor without having to stop. I struggled to read to them and to speak in full sentences. For 38 years, I have watched and felt cystic fibrosis slowly take away my lung function and time with my family and my ability to be the person and parent that I truly want to be. I have been fortunate to be able to try 3 different modulators over the past 5 years. I was involved in the phase 3 study for Orkambi, which ultimately I had to stop because of side effects coupled with a rapidly declining lung function. I then waited for Symdeko with high hopes that this would be the one that would give me back my lost lung function, make life a little easier, and allow me more time with my family. While it initially provided a little bump, it did not last, and I still had the thick, sticky mucus, 2 to 4 hours of treatments every day, IV antibiotics, and fear. Fear that the next exacerbation would be the one that would either require a transplant or take my life. And then came Trikafta. 287 days ago, I took my first dose within minutes, it began working. I was experiencing what has affectionately become known as the purge. And for the next 2 days, I would continue to cough up mucus that I didn't know I had. My lungs would crackle and pop as mucus was finally thin enough to leave. This was a new sensation for me, as up until this point, when I would feel and hear those same sounds, it usually meant that my lungs were bleeding. But this time it was different. For the first time in my life, my mucus no longer reminds me of Slimer from the Ghostbusters. Instead, it is thin, clear, and when it is present, which is not very often, it comes out easily. My lung function has risen to the highest it has been since the beginning of 2015. I run with my kids, exercise harder, and am finishing those projects around the house that I have put off. Even my voice sounds different. All because I can take a deep breath. The numbers do not show you the joy, the happy tears, the sweat, and the love that comes with the simple fact that I now can breathe a little easier. They do not show you the hope that my family has for a future where I grow older and grayer with my wife, where I get to witness my children grow up and accomplish these milestones that so many take for granted. This is the impact beyond the numbers that Trikafta has had on my life and on so many others. This is an impact that we need to foster and make sure that all those with CF get to experience. Thank you again for your time, and may we all breathe in hope.
Well, thank you for providing your direct experience with the medication. I don't know if I don't see any comments at the moment directly. But I think we do have Lori Rogers available. Is that right? I'd like to fit in your presentation next before the break. So Laura Rogers is also a parent of an individual with cystic fibrosis.
Are you able to hear me?
Yes, but with an echo. Maybe there's 2 microphones being used. Maybe try one more time. And I think we're still have.
How about How about now?
Perfect.
Good, that sounds good.
Better? Okay. Hello, I'm Lori Rogers, and I do have an additional conflict to add. I do serve in 2 volunteer positions with the CF Foundation. First, as a board member of the Minnesota Dakotas chapter, and secondly, as a member of the Compass Advisory Board. I'm from Deadwood, South Dakota. My husband and I have been married for 25 years and have raised 4 children, including our youngest, Madeline, who is 17 and will be starting her senior year of high school next week. Madeline arrived 7.5 weeks early and started the first 2 weeks of her life in the NICU. At just over 1 month of age on May 19th and seemingly healthy, we received news that one of Madeline's newborn screenings had indicated the possibility of cystic fibrosis. It was nearly a month before we had an official diagnosis, and we soon made our first trip of many, the 350 miles across the state to meet Madeline's care team, who have all become dear friends over the last 17 years. In 2003, when Madeline was born, the life expectancy for a child with CF was 32 years old. We were helpless, we were mad, and we were scared. Although she has had to endure some hefty antibiotics several times through IV for respiratory infections, her biggest battle has always been her digestive system. We tried many strategies to maintain her weight and decrease the amount of daily stomach discomfort she was experiencing. Madeline also experienced several hospitalizations over her first 14 years related to this digestive function. Ultimately, we made the decision for Madeline to have a feeding tube placed. Although primarily to benefit her weight, it was also to assist in administering the medications for management of her persistent, unpredictable issues. Despite these challenges in comparison to some others with CF, Madeline has been lucky. She has maintained her health enough to experience life like many kids, albeit with some restrictions and a lot of extra work on her part. She has always gone to public school, been on the honor roll, has a great group of friends, she hunts, has sleepovers, and been a 3-sport athlete throughout most of her high school years. In 2015, when Madeline was 12, she was eligible for Orkambi. Although her health was well maintained with this modulator, we were still eagerly awaiting some of the therapies in the CF pipeline. In the summer of 2018, Madeline was enrolled in the triple trial. After the initial 8 weeks, which was in early September, that involved possible study drug or placebo, she was officially switched to AKA the triple. While in this trial, the clinical outcomes and measurements are kept confidential, even from the patient family. So we really had no idea what, if any, measurable clinical effect the drug might be having. By January of 2019, at our 10th research visit, we were to have a routine CF clinic visit as well. Matt had had a good winter, no illness or anything adverse to report. Finally, we would get some feedback and some numbers to look at after 4 months. To our surprise, Madeline's weight was up 2 pounds and her lung function was over 100%. This was an increase of 14% in 4 months, numbers we had not seen since she was age 4 or 5. It was about this time, with nearly a year of monitoring and no use, her feeding tube was removed as well. At this point, I started to pay even more attention to all her symptoms. After returning home from this visit, Madeline developed a cough. She increased treatments, got extra rest, and in about 3 days, the cough was gone. Madeline had never in 16 years gotten over a cough without antibiotic treatment. I also noticed that her sleeping position had changed. Many mornings she would be asleep on her back with her knees bent and drawn up to her stomach with her feet flat on her bed. She always said her stomach was just more comfortable when she slept in this position. For years, even in her sleep, she was managing her CF. I now noticed she was no longer sleeping with her knees bent. Instead, she looked relaxed. Not only had her sleep improved, her mood was noticeably improved. Her family and friend relationships improved. One of her sisters even speaks of this regularly. And even close friends and family have commented how much Madeline has really grown up over the last year or so. This very much could be true, but we all believe she has lost some of her edginess simply because much of her discomfort and fatigue has finally been relieved. I don't know if any of us, including her, ever knew how affected her mood and personality had been. Since Trikafta's approval, Madeline is still participating in a rollover study and has now been taking it for almost 2 years. She feels great, her measurable symptoms are improving, and has only had the need for antibiotics 1 time during this time. I don't think Madeline has ever thought CF held her back. That being said, she is at the age that post-high school plans are needing to be made. She has always wanted to further her education, and for a long time she has said she was going to stay close to home to attend college. Over the past year, those plans have changed, and she now hopes to at least move across the state for school. Maybe it's just her maturing, but because of her comments, I really think she is more confident in her health. She is looking forward to planning her future, knowing that her health has become more manageable, quite possibly even decreasing the number of medicines she takes each day and or decreasing the hours she spends doing treatments. Madeline has had her dreams and future plans reassured by Trikafta. She knows she has a long, bright future, and many others in the CF community have literally gotten their lives back. I don't know how anyone can put a price tag on that. In 2017, the median life expectancy of a child born with CF had grown to 47 years old. That is a 15-year improvement over the course of Madeline's short life. It has been said that CF is the best story in medicine. Madeline and our entire family are living by that statement. There is no doubt in my mind that there will be a cure in the near future, near enough that Madeline could very much benefit from it. If Trikafta is the answer to assisting her and many more in sustaining and improving their health until that day comes, then there should be nothing standing in their way. Thank you.
Well, thank you for your personal experience, and I'd like to thank all the panelists for sharing the information with the staff. At this point, before we break, I want to see if Dr. Person has any comments he'd like to make to the panel so that we're ready to start right at 11 a.m. proper for the next stage. Steve?
All right, thanks. I may invite Matt to do some of this, but just wanted to thank all of the folks who made public comments. In some ways, I guess it's easier for some people to do it via Zoom because it means you don't have to travel to wherever we're holding the public meeting that day. But your insight, your experience, and your testimony obviously is very important for everybody to hear. I do have to say, I think I remember Chad coming to our last meeting on CF, and I hope I'm not mistaken. I think you absolutely, as you said, your voice sounds different, and it's fantastic. So congratulations, it's marvelous. Just before we break, we're going to come back at the hour, and just to make sure that all the panelists are prepared for the voting process that starts at that point, let me ask Matt again to just mention what they need to do to make sure that they're still logged in. Matt?
Thanks, Steve. So voting council members, you should still be logged into the voting system from our pre-meeting session. But if not, during the break, please message myself or Maggie Hool, and we'll help you get back on. And then we would also ask that CTAF members and members of the expert roundtable return a few minutes early before the break ends so we can get started right at 11 a.m. Pacific. So I think that's all I have, so we can move into the break.
Great. See you all back at the hour.